A single cell contains the complete set of chromosomes. In most cases, every gene has two copies, one from the mother and the other from the father. However, the copy number of some genes may not always be 2. They can be 0, 1, or 3, or 4, or more. Traditionally, the copy number is determined by referencing another gene, which we assume to be 2 copies. This assumption does not hold for most of cancer cells and some of the normal cells. How to determine the absolute copy number of a gene or the absolute copy numbers of all genes in a genome has challenged scientists for decades. Now, this problem is solved by a novel single-cell library methodology known as barcode in genome technology, short for BIG, by the scientists in Digenomix. Here's how the process works. A single cell is placed into a small test tube. Once in the tube, histone proteins are removed from genes, making the genes accessible to molecular manipulations. Engineered transposomes are then used to randomly fragment the genes and attaching primers to the resultant fragments at the same time. Although unnoticed at this time, a molecular barcode has been put into each fragment of the single-cell genome by BIG technology through 2 important features within the fragmentation process. First, the breaking points are randomly distributed. Alleles of identical sequences will generate completely different arrays of fragments. In our case here, 2 alleles generated 2 different arrays of fragments. Second, the end of the previous fragment is followed by the start of the next fragment. As we will see, the start and endpoints of each fragment will be utilized in the later bioinformatic process. Primers that have been attached to fragments are utilized to amplify fragments. During amplification, the number of fragments increases, while the sequence of the fragments stays the same. Then, the fragments are sent to sequencing. After sequencing, the sequence of each fragment is mapped to the reference genome. Differing from the conventional bioinformatic approach, we retrieve the information of the start and endpoints of each fragment and register them as 2 elements of the molecular barcode, which is termed as Unique Fragment Index, short for UFI. Fragments with identical UFI are grouped as 1, as they are duplicates in amplification. At the end of this process, 2 alleles of gene are reassembled back by the bioinformatic exercise. As you may have noticed, we got back the exact same number of allele that we started with. If we started with 3 alleles, we will reassemble back 3 alleles at this point. To sum things up, by using BIG technology on single-cell genomes, we can determine the absolute number of alleles without any presumption. 2 key factors are absolutely required to achieve the absolute determination. First, single cell and only single cell. Second, random fragmentation. For more information regarding BIG or our process, please feel free to reach out to us.